We do subunit vaccines, and there are lots of different types of subunit vaccines, but in general, a subunit vaccine takes a piece of the pathogen. It could be a virus or a bacteria. In our case, we only do bacterial subunit vaccines, and they're called conjugate vaccines. My name is Christian Harding, and I'm the Chief Scientific Officer and co-founder of Omniose. We were formerly known as VaxPneumo and rebranded in 2022 when we announced an expanded leadership team. So we have a corporate presence in Boston. That's where the CEO is and the chairperson, and all of our R&D, research and development activities are located here in St. Louis. So we have multiple bacterial vaccines in our developmental pipeline. We have multiple NIH grants, that's the National Institutes of Health, so taxpayer dollars fund a large majority of our research and development activities. One on Klebsiella. In Klebsiella, no approved vaccines yet, high clinical need, and so we see an opportunity to really provide a big societal benefit because there's not products there already, and we have a really compelling product and platform to address in Klebsiella. So that's a $3.2 million grant to really go through and understand, do the vaccine discovery. Which vaccine target of Klebsiella will be the best for the vaccine inclusion? All bacteria, or most bacteria, make a sugary capsule or shield around them that protects them from our immune system and environmental threats. That sugary shield is also a fingerprint. It's a bacterial fingerprint, and it separates them from us. And when we take that subunit, that sugary subunit, and attach it to another immunogenic protein, we get what's called a conjugate vaccine. So again, these are subunit vaccines, and within that, the subunit is two parts. We have a part of the bacterial shield, that carbohydrate or polysaccharide, and we have a carrier protein. And they need to be glued together or covalently linked. You can do that lots of ways. You can purify this and do it with chemistry. You can do multiple different types of precision chemical conjugation. We do it enzymatically. So we've engineered the lab-safe bacterium called E. coli, which every lab works on around the world. It's the workhorse for biological development. So we've engineered E. coli to make that vaccine-specific polysaccharide. We've also said at the same time, make the carrier protein, and what we have is a proprietary enzyme that moves the polysaccharide onto that carrier protein and covalently attaches them. So it's a one-step reaction to make that conjugate vaccine. And that's what differentiates us from Pfizer, other big pharma that use this chemical conjugation approach.